Hi friends, welcome to Applications Corner. In today's video, I'm gonna discuss with you male reproductive system. So it's our anatomy class 1. So if you are new on this channel, then please subscribe to this and so that you can get the latest updates and the latest lectures. So let's start. So first of all, we have to see that what the reproduction actually is. So reproduction is actually an essential function in all living things, and this actually life continues from one generation to another generation by this process. So this is the general definition of reproduction. So now if we move to the next from a general information is given here, like from 300 million to 400 million sperm in each ejaculation, and from them only one can fertilize an ovum. So it's actually a huge amount, but only one is going to fertilize an ovum. So now moving next, the functions of the male reproductive system we are going to see here. So the first function of the male reproductive system is actually the spermatogenesis, and this is actually the major function of this system. And spermatogenesis is actually the production of reproductive cells, and that we call sperms, and they are actually generated from the spermatogonia. So the next function of the male reproductive system is copulation, and copulation is actually the process in which the sperms are transferred to the female and etc. And the third function of the male reproductive system is production of hormones. The next thing is at which time period the puberty starts. So now it begins usually between 9 to 14 years of age, and if the puberty occurring before this age, or it is early than this, then this will be known as precocious puberty, and if it is delayed, then this condition is known as eugonadism. And there is a burst of hormones occur at this age, and this is the puberty stage at which all the things are actually going towards a secondary development. So now we are going to talk about the general physical changes occur at the time of puberty, and whenever there is a changes are occurring. So first of all, we have the enlargement of the external and internal genitalia, and there is the voice changes, there will be the hair growth, then mental changes in the body and body conformation and skin. Next we have sebaceous gland excretions and thick and increase like acne. So there will be all the conditions will be there. The next thing is at which time period the puberty starts. So now it begins usually between 9 to 14 years of age, and if the puberty occurring before this age, or it is early than this, then this will be known as precocious puberty, and if it is delayed, then this condition is known as eugonadism. And there is a burst of hormones occur at this age, and this is the puberty stage at which all the things are actually going towards a secondary development. So now we are going to talk about the general physical changes occur at the time of puberty, and whenever there is a changes are occurring. So first of all, we have the enlargement of the external and internal genitalia, and there is the voice changes, there will be the hair growth, then mental changes in the body and body conformation and skin. Next we have sebaceous gland excretions and thick and increase like acne. So there will be all the conditions will be there. We are seeing here the external genitalia. So external genitalia are actually all the organs or all the parts or all the specific portions that are actually outside the body. So here we have the gonads like testes in the case of male, because we are studying male reproductive system here. So next, if the gonads or testes are undescended by birth, means there is no proper appearance of the testes, then we will say that this is cryptorchidism. So next thing is scrotum. We have scrotum is just like this, and next we have the penis, and penis is like this. So now we see what are the internal genital organs actually. So we have epididymis, then we have vas deferens, and then we have accessory glands. So now moving towards the scrotum. So first portion we are going to discuss here, that is actually from the external genital organ, that is actually scrotum. So scrotum is actually a sac of skin and superficial fascia that hangs outside the abdominopelvic cavity containing paired testicles. So actually the scrotum is actually a sac of skin in which the testicles are present. So next is the structure, that is spermatic cord. Spermatic cord actually encloses the nerves and blood vessels, and next are the testes. So testes are kept 3 degrees Celsius lower than the core body temperature needed for the sperm production, and they are controlled by the different muscles. So we have to make sure that the temperature of the testes are actually keeping at lower than the body's temperature, and that is up to 2 to 3 degrees Celsius lower than the body's temperature, because this temperature is actually very much specific or very much suitable for the production of sperms in the seminiferous tubules. So next we are going to see that the temperature that is controlled. So the factors involving in this mechanism. So we have cremaster muscle. The cremaster muscle is actually functioning as like we see, when it is cold, it contracts, pulling the testes up towards the core of the body, and whenever there is a cold outside, then the cremaster muscles will become like they contract and they pull the testes up towards the core of the body, so that the temperature is not going to too cold or too hot. So next, when it is warm, then these muscles relax, allowing the testes to descend away from the body. So whenever there is the warm conditions, and then they will be relaxing, and then they will be moving the testes descend away from the body, means that testes will be descend down. A vertical septum of the subcutaneous tissue in the center divides into two parts, each containing one testis. So there is actually a tissue that is actually dividing the scrotum into two portions, and there is a vertical septum, and it divides the scrotum into two portions, and each one contains one testis. So next, smooth muscle fibers called the dartos muscles in the subcutaneous tissue and contract to give the scrotum its wrinkled appearance. So the wrinkled appearance, you see in the diagram that there is a wrinkled appearance of the scrotum, and this wrinkled appearance is actually due to the dartos muscle, and the dartos muscles are present in the subcutaneous tissues, and whenever they contract, they give the wrinkled appearance to the scrotum, and when these fibers are relaxed, the scrotum is smooth. So this is the condition. Whenever they are relaxed, then they make the scrotum smooth. Next, moving towards the scrotal structures, and like you can see here, this is the spermatic cord, and like here, and here we have epididymis. This is the epididymis, and that is coming from the testes, and here we have the testes, and here we have scrotum. So this is the scrotum, that is a sag-like structure enclosing the testes. So there will be the vertical septum on the other side, and we have in the scrotum, that is dividing into two portions, and each containing one testis. Now the testis is an oval structure about 5 cm long and 3 cm in diameter, and the testis is covered by a tunica albuginea tissue layer, and then the testes are located inside the scrotum, and there are about 250 lobules in each testis, and each contains 1 to 4 seminiferous tubules that converge to form a single straight tube which leads into the rete testis. So now what we have, actually there are 250 lobes in each testis, and these lobules actually contain 1 to 4 seminiferous tubules. Seminiferous tubules are actually the tubules which are producing the sperms, having the lytic cells and having different cells, nutritional cells, and we have, so next that converge to form a single straight tubule, and these 1 to 4 seminiferous tubule combine to form a single straight tubule, and that will lead into the rete testis, and then from the rete testis they will be going into the epididymis. So there is a short efferent duct exit the testis, and next are the interstitial cells. These are the cells of Leydig which produce the male sex hormone and are located between the seminiferous tubules within a lobule. So first of all, you can see that this is scrotum, the layer that is containing testes, and then we have seminiferous tubules here. You can see that these are the seminiferous tubules. The nexus of the seminiferous tubules is over there. Next we have the scrotal skin. This is the scrotal skin, means the skin portion that is actually covering the scrotum. Next we have vas deferens. This is the vas deferens coming from the epididymis. So this is the epididymis. This epididymis arises from here. This is rete testis area. This is vasa efferentia, and from the vasa efferentia the sperms are going to the epididymis, and then from the epididymis they are going towards like this, and then moving towards the vas deferens, and then from the vas deferens they are going towards the other tubules. So here we have the sagittal section of the testis and epididymis. So this is, first of all, we have septum, that is septum is separating the testis. Next, this is the lobule, and we have tunica albuginea that is covering the testis, and then seminiferous tubules, like this is the nexus of seminiferous tubules. Then straight tubule that is arising from many seminiferous tubules joined to form straight tubule that will open into the rete testis. Here we have the rete testis, and next there is a efferent duct. This is the efferent duct, and that will be in the epididymis, and then from the epididymis, same case as the dextrous deferens. Next we have the epididymis, and the epididymis is actually a portion and head. This is the head of the epididymis arising from the testis. This is the body of the epididymis that is alongside the testis, and this is the tail of the epididymis that is here, and this is going towards the ductus deferens. Next we have some microscopic view of the seminiferous tubule, and this is the histology. Now this is, you can see that this is the lumen of seminiferous tubule, and this is spermatozoa. You can see here these are the different thread-like structures you can see here, and these are actually the haploid cells, and these are the sperm cells. Next you can see here the spermatid. They are having different structures, and like this is secondary spermatocyte here, this is the secondary spermatocyte, then primary spermatocyte, and here is a spermatogonium from where all the generations will be there. So this is the basement membrane we have. This is basement membrane. Next, interstitial endocrinatocyte. This is the Leydig cell. The Leydig cells are producing the sperms actually. See that how the testicular functions are regulated by hormones. So first of all, the hypothalamus releases the gonadotropin releasing hormone, GnRH, the short form, and this gonadotropin releasing hormone stimulates the anterior pituitary to release the follicle stimulating hormone and the luteinizing hormone. The follicle stimulating hormone causes the subtesticular cells, means the nursing cells, to release the androgen binding protein ABP and inhibin. Now luteinizing hormone stimulates the interstitial cells to release the testosterone. So this is the major factor, the GnRH, and the GnRH, the gonadotropin releasing hormone, is going to stimulate the different cells to produce the androgen binding protein and inhibin, and alongside there is a testosterone also. We have ABP. This is the androgen binding protein, and this is the binding of the testosterone enhances the spermatogenesis. Next we have secondary sex characteristics are actually developed by the testosterone. Next we have epididymis. Epididymis, a long tube about 6 meters located along the superior and posterior margins of the testis, and sperm that leave the testes are immature and are incapable of fertilizing the ova, so they are completed with their maturation process and become fertile as they move through the epididymis. So just like mature sperms are stored in the lower portion and or the tail, so of the epididymis. So we see that there is a head of the epididymis, and then this is the body, and there is the tail of the epididymis, and here we have the testes just like this. So this is, we have epididymis. Next is the spermatic cord. Spermatic cord contains the proximal ductus deferens. There is a testicular artery and veins, lymph vessels, testicular nerves, cremaster muscles, and connective tissue coverings. So these are all the portions, these are all the parts that are actually exhibited by the spermatic cord. Next is the duct system. Sperm cells pass through a series of ducts to reach the outside of the body. After they leave to the testis, the sperm passes through the epididymis, then to the ductus deferens, then through the ejaculatory duct, and then through the urethra. Next we have ductus deferens, and this is also known as vas deferens or the vasa deferens. So this is actually a fibromuscular tube that is continuous with the epididymis and enters the abdominopelvic cavity through the inguinal canal and passes along the lateral pelvic wall, behind the bladder and towards the prostate gland. Just before it reaches the prostate gland, each ductus deferens enlarges to form an ampulla. So sperms are stored in the proximal portion of the ductus deferens and near the epididymis. Next is the ejaculatory duct. We have each ductus deferens at the ampulla joins the duct from the adjacent seminal vesicles. We know that there is a pair of the seminal vesicles, one of the accessory glands, to form a short ejaculatory duct. And now each ejaculatory duct passes through the prostate gland and empties into the urethra. Next, we have urethra that extends from the urinary bladder to the external urethral orifice at the tip of the penis. It is a passageway for sperm and fluids from the reproductive system and urine from the urinary system. It is divided into 3 regions: the prostatic urethra, then we have membranous urethra, and then the penile urethra, also called spongy urethra or the cavernous urethra. So these names are actually due to the presence of different structures, different tissues there. So we have named these structures and like this. So next is the ejaculatory duct. We have each ductus deferens at the ampulla joins the duct from the adjacent seminal vesicles. We know that there is a pair of the seminal vesicles, one of the accessory glands, to form a short ejaculatory duct. And now each ejaculatory duct passes through the prostate gland and empties into the urethra. So here we have an overview of this whole system. So first of all, we will see that this is the ureter and then trigone of urinary bladder. We have, this is the trigone and then seminal vesicle here and opening of ejaculatory duct. This is the ejaculatory duct coming from this side, and if we start from here, this is the corpus spongiosum and opening of bulbourethral. Next, if we talk about the urethra, extends from the urinary bladder to the external urethral orifice. Next, we have urethra that extends from the urinary bladder to the external urethral orifice at the tip of the penis. It is a passageway for sperm and fluids from the reproductive system and urine from the urinary system. It is divided into three regions: the prostatic urethra, then we have membranous urethra, and then the penile urethra, also called spongy urethra or the cavernous urethra. So these names are actually due to the presence of different structures, different tissues there. So we have named these structures and like this. Next are the some accessory glands and next are the accessory glands. Accessory glands are the seminal vesicles, next prostate gland, and the bulbourethral glands. Accessory glands are actually glands which are actually providing the different fluids, different chemicals, or supporting the cells to produce the sperms and hold the reproduction and hold the production of spermatogenesis. So like that. Next are the accessory glands. Accessory glands are the seminal vesicles. They include prostate gland and the bulbourethral glands. These glands secrete the fluids that enter the urethra, so they actually nourishes, they actually provide the hormones to the secretions. So first of all, there is a seminal vesicles. The glands posterior to the urinary bladder, these are the seminal vesicles, and each has a short duct that joins with the ductus deferens that is coming from the testes at the ampulla to form an ejaculatory duct, which then empties into the urethra. So what we have, the ductus deferens is coming from the testes and it joins with the duct from the seminal vesicle at the ampulla, and then they will coming towards the ejaculatory duct. Then the fluid is viscous and it contains the fructose, prostaglandins, and different proteins. Next we have the prostate gland. Now prostate gland is a firm, dense structure about the size of a walnut that is located just inferior to the urinary bladder. So it is present inferior to the urinary bladder. It encircles the urethra as it leaves the urinary bladder, and numerous short ducts from the prostate glands empty into the prostatic urethra, and the secretions of the prostate are thin, milky, colored, and alkaline. They function to enhance the motility of sperm. So this is the major function of the secretions of the prostate gland, that they enhance the motility of the sperms. So next are the bulbourethral glands. They are also known as Cooper's glands, that is named after the scientist. So next we have, they are small, about the size of a pea, and are located near the base of the penis. A short duct from each enters the proximal duct of the penile urethra. So now in response to the sexual stimulation, the bulbourethral glands secrete an alkaline mucus-like fluid. So this fluid will be acting as the supporting fluid for the sperms to moving to their location. We have seminal fluid or semen, a slightly alkaline mixture of sperm cells and secretions from the accessory glands, and they actually make the whole semen. So secretions from the seminal vesicles make up about 60% of the volume of the semen, and with most of the remainder coming from the prostate gland, and the sperms and secretions from the bulbourethral glands contribute only a small volume. The volume of the semen in a single ejaculation may vary from 1.5 to 6 milliliter. There are between 50 to 150 million sperms per milliliter of the semen, and if this count is below than 10 to 20 million per milliliter, then usually it represents the fertility problems. So there may be the different fertility problems will be there if the sperm count is less than the determined value. Next we have the penis. Penis is actually a cylindrical pendant organ and located anterior to the scrotum and functions to the transfer of sperms to the vagina. Now it consists of three columns of erectile tissues and that are wrapped in connective tissue and covered with skin. And what are these tissues? So the two dorsal columns, there are two dorsal columns are the corpora cavernosa, and the single midline ventral column surrounds the urethra and is called corpus spongiosum. So actually we have two distal columns and we have one single midline ventral column. The distal columns are two columns, they are known as corpora cavernosa, and they are actually erectile tissues. And number third one is the corpus spongiosum, and this corpus spongiosum is actually covering the urethra, it surrounds the urethra. So next, the penis is actually having three parts: a root, then a body, that is shaft, and glans penis. The root of the penis attaches it to the pubic arc and the body is visible pendant portion. Next we have two corpus cavernosum penis, and the corpus spongiosum expands at the distal end to form the glans penis. The urethra, which extends throughout the length of the corpus spongiosum, opens through the external urethral orifice at the tip of the glans penis, and a loose fold of skin called the prepuce or foreskin covers the glans penis. So here you can see that we have scrotum, we have discussed earlier. Then we have external urethral orifice here, and this is the urethra. And next we have, this is the glans penis, there is a tip of the penis known as glans penis. Then we have spongy urethra because its name from the corpus spongiosum. So this is the spongiosum layer that is covering like this, and this is surrounding the urethra, and this is the spongy urethra here. Then corpora cavernosa here, this is the corpora cavernosa, and there are actually two bands present here. Next we have membranous urethral portion, this is the membranous urethral portion. Next we have bulbourethral glands here, bulbourethral gland that is present actually a base of the urinary bladder or the base of the urethra. Next we have prostate urethra, then we have urinary bladder and prostate gland located here. So these are the different parts present here. So here we have the internal structure of the penis. So first of all, we will see that there is a corpora cavernosa, two bands of the corpora cavernosa. These are the two bands. And the next corpora cavernosa, this is actually the same bands moving along the whole length of the penis. So next we have the glans penis here. There is a tip of the penis and urethral opening is also here. So this is the urethra you can see here, and the corpora spongiosum is actually covering the urethra from this side. So this is the corpus spongiosum. This is covering the urethra. So this is some microscopic view of the whole penis, and this is the epididymis we have, then tunica albuginea of corpora cavernosa, and this is the whole next corpus cavernosa. This is the corpus cavernosa here. There are the two bands. Then median septum is here. This is the median septum. Then corpus spongiosum. This is the corpus spongiosum. And then this is the cavernous urethra we have. This is the urethra, and so the corpus spongiosum is covering the urethra, surrounding the urethra. Then tunica albuginea of corpus spongiosum is also here. And then this is all the microscopic view of the penis. Next we will talk about the erection. So erection involves the increase in length, width and firmness of the penis. So next, changes in the blood supply, then arterioles dilate and veins constrict. So there are two different scenarios. Arterioles will dilate and veins will constrict, and the spongy erectile tissue fills with the blood. The erectile dysfunction is actually a dysfunction or the malfunction, and this is also known as impotence. Next are the hormones. The follicle stimulating hormone stimulates the spermatogenesis, the process of producing the sperms. Interstitial cell stimulating hormone ICSH, this is stimulating the production of testosterone. Then we have testosterone stimulates the development of the male secondary sex characteristics and spermatogenesis. Spermatogenesis is actually a process of producing the sperms, and now sperms are produced within the seminiferous tubules we have discussed earlier, and the interspersed within the tubules, and they are the large cells which are the testicular cells. These are the Sertoli cells, and which support and nourish the other cells. They are also known as nourishing cells or nursing cells. So early in the embryonic development, the primordial germ cell enter the testes and differentiate into spermatogonia. Now spermatogonia are diploid cells, and each with 46 chromosomes to 23 pairs, and located around the periphery of the seminiferous tubules. At puberty, hormones stimulate the cells to begin dividing by mitosis. So some remain at the periphery as spermatogonia. Others become primary spermatocytes because they are produced by mitosis, and primary spermatocytes like spermatogonia are diploid and have 46 chromosomes. Each primary spermatocyte this undergoes through the first meiotic division, meiosis 1, to produce two secondary spermatocytes, each with 223 chromosomes, and they are haploid in number. Just prior to this division, the genetic material is replicated. During meiosis 1, one chromosome goes to each secondary spermatocyte. In the second meiotic division, meiosis 2, each secondary spermatocyte divides into produce two spermatids. There is no replication of genetic material in this division as was present earlier, but a single stranded chromatid goes to each cell. As a result of two meiotic divisions, there is each primary spermatocyte produces, and this is 4 sperms production occur. So now each spermatid, there are 4 spermatid production will be there. So each spermatid has 23 chromosomes, haploid in number, and one from each pair. In the original, there is a primary spermatocyte. So this is the whole spermatogenesis, and this is related with the cells. So we have Sertoli cells here, and now here's a spermatogenesis occurring. So here you can see that spermatogonium diploid in number, and this is the spermatogonium. Now this is by mitosis, it is converted into the primary spermatocyte, diploid in number. Then by the meiosis 1, the primary spermatocyte is going to the secondary spermatocyte, that is haploid in number, having 23 chromosomes each. So now these two secondary spermatocytes by the meiosis 2, they are converted into the spermatids, and these spermatids are actually 4 in number, and each secondary spermatocyte produces 2 spermatids. Now these spermatids will actually nourish, and they will be moving towards the sperms by different adaptations, by different modifications. This is the structure of the sperm we have. So this is the head of the sperm, then this is the middle section from this to this length, and this is the tail known as flagellum because this is flagella like. So this is the mammalian sperm gamete. Now we have acrosome here, that is at present at the tip. Then we have cell membrane, then there is a nucleus here, then there is a centriole here, then mitochondria will be here for the production of energy. Here is another elaboration of spermatogenesis, like this is testes, then spermatogonium to primary spermatocyte, then primary spermatocyte to 2 secondary spermatocytes, and then from each secondary spermatocyte there is a 2 spermatids each, and then these spermatids are actually modified to sperms. So here is an overview of the path of the sperms, that the paths from where the sperms are actually moving. So first of all, they are in the testes, production occur, then moving towards the epididymis, then from the epididymis they are into the vas deferens, then to the seminal vesicles, and then from seminal vesicles secretions there is a prostate gland secretions, then moving towards the sphincter valves, and then they enter into the urethra, and then they ejaculated out of the body. So friends, this was all about our today's lecture, and I have discussed with you the male reproductive system, and this was all about the lecture. So I hope you will understand whatever I made the presentation to you. So see you very soon in the coming video.